Bismillahirrahmanirrahim, and welcome back to this next video. And this is the fourth video in the series of videos on immunology. In my previous videos, I've told you about the evidences that proves that there is an immune system in the living organism and that the immune system protects the living organism from different pathological conditions. I gave you an overview of the important components of the immune system, that which are types of the cells, organs, tissues and substances that are involved in the immune system, and that the immune system using those particular components provide immunity to the living organism. In my last video, I started on the history or the birth of immunology, and I gave you some of the important events that occurred in the history that marked the beginning of the immunology. Now, I'll continue the history, and when you talk about the history of immunology, you cannot ignore the contributions of the Edward Jenner. Now, Edward Jenner, he made a very important observation, and what Edward Jenner observed was that those particular milkmaid who have contracted the mild cowpox disease, because the milkmaid, they are dealing with the cows, so they somehow contracted the mild cowpox disease. Now, this cowpox disease is a skin condition in animal, and this cowpox is caused by a virus from the orthopox virus genus. So he observed that those particular milkmaid who have contracted the mild cowpox disease, they were then protected from the severe smallpox condition. That means that the cowpox have somehow made them immune to the smallpox virus. So his hypothesis was that if we can use the fluid from the cowpox pustules, that is going to give immunity to individual against the smallpox. Now, this was a great question. This was a great idea. So how he tested this idea? What he did was that he inoculated an 8-year-old boy with the pustules, the cowpox pustules, and then he intentionally infected that 8-year-old boy with the smallpox virus. So he first inoculated the 8-year-old boy with the cowpox pustule to test his hypothesis that this inoculation is going to make this 8-year-old boy immune to the smallpox, and then he intentionally infected the boy with the smallpox to test his hypothesis. And it worked. This particular idea that cowpox protects against the smallpox condition, it is proved by this particular experiments. This particular idea worked, but of course you cannot do this in the current day because of the medical ethics, because the ethics are that you cannot directly use a vaccine, directly use a drug on the human beings unless you have success in the lab and you have success in the animal models. But somehow at that particular time, the medical ethics, I believe, was not that much a big issue. So he did these experiments on this 8-year-old boy. But the good thing about this experiment was that this succeeded, it worked, and it paved the way for other experiments in the immunization program. Now, the success of the Edward work, it spread throughout the Europe. And this success actually led to the other experiments, other historical experiments in the field of immunology to give boost to this subject of immunology. Then come the era of the Louis Pasteur, the great Louis Pasteur in 1877. He actually worked with the cholera. This cholera is actually an acute diarrheal disease. And this cholera is caused by a bacteria which is known as the Vibrio cholera. So he was testing this idea of immunization and vaccination on this cholera disease. What he did was that he grew the culture of the Vibrio cholera in his lab, and to confirm this, that the culture that he has grew in the lab, that is Vibrio cholera, he inoculated this culture into the chickens. The chicken, they contracted the diarrheal disease, thereby confirming that the Vibrio cholera was the organism that he grew in his lab. He went on his summer vacation, and when he came back from the summer vacation, he used this old bacterial culture again to inoculate the chicken. But to his surprise, the chicken, they became ill, but they survived this old bacterial culture. Now this was something of surprise, because the same culture that was causing cholera in the chickens, but the old bacterial culture that was not causing that much severe cholera in the chickens, that they survived that particular condition, but they became ill, but they survived the condition. So that was a good thing. That was, you can say, that led to some other questioning in the field of the vaccination, that why this is happening. The fresh cultures, they are working, the old cultures, they are not working. There might be something surprising, there might be something fascinating in this concept. What he did was he made some more fresh cultures, and now he did two sort of experiments. The first experiment that he did with these fresh culture was that he inoculated them to the fresh chickens. By that I mean that those particular chickens had never been inoculated with the Vibrio cholera. So this fresh culture, when that was inoculated to the fresh chickens, all of the fresh chickens, they died. But when he infected this fresh culture to the previously infected chickens that were inoculated with the old bacterial culture, they survived the condition. What this means is that this old bacterial culture has somehow developed some immunity in these chickens, that they are now resistant to the fresh culture of the Vibrio cholera, and they are surviving the condition. They are not getting infected with the cholera. Now, what he did was that he used a term like the attenuated. He was not using this term attenuated, but these days we are using this term attenuated. And attenuated simply means that the pathogenic organism has somehow lost its pathogenicity. It is still activating the immune system, but it is not causing disease in that particular organism or in the human beings. So this old bacterial culture, as that was attenuated, he said that the age has made these bacterial culture attenuated. Now they are not that much infective. So he used this term vaccine for these old bacterial cultures, and he said that these old bacterial culture can be used as a vaccine to protect the chicken from the cholera. Now why he called this as the vaccine? Now this was a good question, why we call the vaccine as a vaccine. Now this vaccine, it is actually derived from a Latin word vacca. This vacca means cow, and he called these attenuated old bacterial Vibrio cholera cultures as vaccines because he wanted to give he gave the name vaccine in honor of the Jenner work, because Jenner, he worked with these cows because he was actually collecting the cowpox pustules from these cows. So he called this vaccine from this Latin word vacca, which means cow, in honor of the Jenner's work. So the term vaccine that was given by this Louis Pasteur at the end of his experiments on the cholera. Now, after the Louis Pasteur, then come the he did some other experiments as well. He performed the experiments on the cholera in 1877. Then he worked on the anthrax in 1881. Now, what he did in this anthrax experiment was that he took some sheep and he inoculated these sheep with the heat-treated culture of the Bacillus anthracis. So the Bacillus anthracis, that is actually the causative organism for the anthrax. He heat cultured these Bacillus anthracis. He inoculated some sheep with these heat treated cultures of the Bacillus anthracis. Then what he did was that he made two groups of the animals. One group was the vaccinated sheep, those particular sheep who have already been treated with this heat-treated culture of the Bacillus anthracis. The second group of the sheep, they were unvaccinated. And the result was that the vaccinated sheep, they survived the anthrax condition. The unvaccinated sheep, they got the disease of the anthrax. And these experiments, they actually marked the beginning of the immunology, that if you are exposed to an attenuated version of a particular pathogen, your immune system is then going to protect you from the real infection of that particular organism. So if you are treating the sheep with the heat-treated culture first, then you are giving them the unattenuated Bacillus anthracis. The heat-treated culture have made these sheep immune to the natural Bacillus anthracis. Therefore, they are not getting the anthrax condition. The unvaccinated the unvaccinated sheep, because they have not been exposed to Bacillus anthracis first, they are not immunized. So when they are exposed to the wild type of the Bacillus anthracis, they are not going to survive the condition, and they are going to get the anthrax. Now, these experiments that actually marked the beginning of the immunology. Now, in 1885, what the Louis Pasteur did, that was the first, you can say, human vaccination experiments that he did in 1885. And what he did was that there was a boy, the name of the Joseph Pasteur. He was repeatedly bitten by the rabid dog, and these rabid dog that actually carry the rabies. And rabies is actually a condition that affects the neurological system, the nervous system of the individual. So Joseph Pasteur, he was repeatedly bitten by these rabid dogs, which carries these rabies virus. What he did was that he made attenuated cultures of these rabies virus. He gave the several shots of this attenuated rabies vaccine to Joseph Pasteur, and Joseph Pasteur, after getting these attenuated vaccines, he survived, and he was not killed by this lethal rabies infection. He survived the condition, and then he served as a caretaker in the famous Pasteur Institute. The Pasteur Institute that was, you can say, inaugurated in 1887. And the Pasteur Institute, in those particular days, that was important for the treatment of the rabies patient. Those who has got bitten by the rabid dogs, that were rushed to the Pasteur Institute, and Joseph Pasteur was actually helping them to get rid of these rabies, so that they do not get infected. Now, one of the fascinating thing about the rabies vaccine is that after you have been bitten by the rabid dog, and you after not a longer period of time, if you get the vaccine, at that particular time, if the virus have not yet reached to your nervous system, the neurological system that has not yet been begun, and if in that particular stage, if you get the rabies vaccines, you are going to survive. So this is one of the fascinating thing about the rabies vaccine. So this Pasteur Institute that even exists now, one of the greatest institute in the world that is dealing with these infectious diseases, in the treatment of the infectious diseases, and in the development of the vaccines. So this is how the, you can say, the fascinating subject of the immunology started, and these are just some of the important historical events that occurred there and that marked the beginning of the immunology. So if you like the video, please subscribe to my channel, hit the notification bell so that you can get notification for all of the coming videos, and share it with your friends. I'll see you in the next video.